Hello, my dear students. Today in this class, we are discussing very important questions from the molecular basis of the inheritance. So this is one of the very important chapter. So according to your blueprint, this chapter carries total 13 marks in your board exam. One of the very important chapter for your board exam. Here, so in this chapter, one MCQ will be asked, one MCQ question. So same 2 mark question and 5 mark questions, that will be 2. Okay, total 10 mark. Total 10 mark questions will be asked from the 5 mark section. Okay, total 10 plus 2 plus 1, that is equal to total 13 mark questions will be asked. Okay, so one of the very important chapter. So in this session we are discussing the MCQ question. Okay, very important MCQ questions. So after that 2 mark question, then 5 mark question. So like this we are discussing the question. So let us start with the first question. In a nucleotide, the nitrogen base is joined to the sugar molecule. So how it is joined? Option A, phosphodiester bond. Option B, glycosidic bond. Option C, hydrogen bond. And option D, A and B. Here, first you should know what is nucleotide. Nucleotide that contain the pentose sugar. So in case of DNA, pentose sugar. Then nitrogen base, which contain the nitrogen bases, that is adenine, guanine, then cytosine. So these are the nitrogen bases: A, T, C, G, adenine, thymine, guanine, cytosine, guanine. So these are the nitrogen bases. So this for this sugar group, nitrogen base is attached. How it is attached? So which bond is present in between that? That is phosphodiester bond? No. The phosphate group attached to the sugar by the phosphodiester bond. But the correct answer is glycosidic bond. With the help of the glycosidic bond, the nucleotide to the in a nucleotide, the nitrogen base is joined to the sugar molecule. Option B is the correct answer. Next question. If a double strand DNA has 20% thymine, the percentage of guanine in a DNA. You know that adenine bind with the help of hydrogen bond with the thymine, cytosine with the guanine with the triple 3 hydrogen bond, ATCG. So here amount of purines, that is equal to amount of pyrimidines. So you know the Chargaff rule, right? So here the double strand DNA has 20% thymine. So here 20% thymine is there. Then what is the percentage of the adenine? So that is also 20%. That is also same. So totally 40% is over. Then in 100%, so it is given in a percentage. So in 100, 40 is 40 is total number of adenine and thymine is 40% and remaining 60%. So cytosine and guanine, so they are same. So 60 divided by 2 to 3. So that is each cytosine is 30% and guanine is 30%, total 100%, right? The percentage of guanine, they asked the percentage of guanine, that is 30%. The correct answer is option A, that is 30%. So to solve this question, you should know the amount of purines, that is equal to amount of the pyrimidines, alva. So that should be same option. So option A is the correct answer. Next question. If a DNA contains 1000 base pairs, what would be its length? You know that the length of the read the question properly. If the DNA contains 1000 base pairs, what would be its length? 1000 base pairs, right? So here 3400 angstrom, 34,000 angstrom, 6800 angstrom, and 1000 angstrom. Okay, here you know that one base pair that is equal to 3.4 angstrom. Okay, so into 1000 base pairs means 1, 2, 3. So that is 3000 and 2, 3. Right. So point here, that is 3400 angstrom. Here, how you should solve this question? You know the value. The length of one base pair is 3.4 angstrom. So 3.4 total, how many base pairs are given? 1000 base pairs. So that is equal to 3, 4, 1, 2, 3, 0. So 3400 angstrom. Option A is the correct answer. Next question. What is not true for a DNA in prokaryotes? Okay, which is a incorrect sentence that you have to find out. The DNA in prokaryotes. Prokaryotes means, for example, bacteria which do not contain the well organized nucleus, right? Example, bacteria. Here, uh, present in the form of compact structure called nucleus. Yes, this is the correct answer. The coils are maintained by non-histone basic proteins. So that is also a correct answer. Found in cytoplasm in supercoiled condition. Yes, they are found in a cytoplasm in a supercoiled condition. Packaged as nucleosomes along with the histones. No, they do not. They are not packaged as nucleosomes and they do not contain the histone proteins. The wrong, the sentence which is not true, that is D. Okay. Next question. Pick the right difference between the DNA and RNA. Sugar and phosphate, sugar and purines, purines and phosphate, sugar and pyrimidine. So you know that DNA is the double strand molecule and RNA is the single strand molecule. But here what they are given, how the DNA and RNA they are different. Sugar and phosphate group, sugar and purine group, purines and phosphate, sugar and pyrimidine. Here, you should know that in RNA, sugar, so that is the ribose sugar will be there. In case of RNA, ribose sugar will be there, but in case of the DNA, that is there will be a pentose sugar. Okay, so the sugar group is different in both DNA and RNA. So you can take this option A, B, and C. Sugar, so that is different in both DNA and RNA. Phosphate group is same both in DNA and RNA, so we cannot take this option. Purines and pyrimidines, adenine and guanine, cytosine and thymine. So adenine and guanine, they are the purines. Cytosine and thymine, they are the pyrimidines. So in case of the RNA, there is a uracil group will be there. In place of the thymine, uracil group is replaced. So these are the pyrimidines. Cytosine and thymine, they are pyrimidines, right? So here the thymine is replaced by the uracil. Okay. So that is the uh, difference. So the correct answer is sugar and pyrimidines, not sugar and purines, sugar and pyrimidines. Next question. Hershey and Chase experiment providing or proving DNA as genetic material was based on the principle transduction, transformation, transcription, translation. Here, Hershey and Chase, they proved the DNA as a genetic material that is based on the transduction principle. But how it is a transduction? They grow a bacteriophage that is able to infect the bacteria, some E. coli bacteria. So they introduced some radioactive substances inside the bacteriophage. So if that when the bacteriophage transfers the genetic material into the bacteria, that process, so that is based on the transduction. So this Hershey and Chase experiment ಅನ್ನ ನಾನು 5 marks ಗೆ ನಿಮಗೆ in detail ಆಗಿ explain ಮಾಡ್ತೇನೆ. ಓಕೆ, it is not a transformation. Transformation and the just it is a transfer from the outside to the inside the um, bacterial cell. Transcription, you know that this is the process of formation of the RNA from the DNA, and translation, it is the process of formation of the synthesis of the protein. So that is the translation. So these three methods are wrong. So the correct answer is transduction. Okay. Next question. A bacterial colony containing DNA made up of 100% N15, so it should be like this, N15 nitrogen bases is allowed to replicate in a medium containing N14 bases. After one round of replication, result would be here. Uh, the bacterial colony, when they are grown in 100% N15, so all the nitrogen bases, they contain the inside the DNA, they have the N15 molecules, right? But when they grown in a medium which contain the N14, they contain the N14. So here also in the next, in after one uh, generation, after one replication, what happens? See here, this is the original DNA, okay? So which is having the N15 nitrogen bases. When it is allowed to replicate in the N14, when it is transported to the N14 medium, then it will be like this. One strand contain the N14 and another strand it contain the N15. N15 anna ಹೊಂದಿರುತ್ತೆ. So after the one replication. So what is the correct answer? Choose the option. All individuals will be identical to the parents. So these are the new individuals, but these are not identical to the parents. All individuals will be hybrids. Only 50% individuals would be hybrids. All individuals would have DNA made up of 100% N14. So this is not true. Only 50% individuals would be hybrids. No. But we can say that all individuals, if it is forming the two DNA molecules, then the two both DNA molecules, how many DNA molecules are formed? So these both are, they are hybrid. Okay. The correct answer is option B. Next question. Cistron is the coding sequence of DNA. The functional unit of DNA molecule that codes for particular gene product. Intervening non-coding sequence of DNA, the sequences which are removed during the RNA splicing. The sequence which are removed at the RNA splicing, they are called as introns. And intervening non-coding sequence of the DNA, which is not a non-coding sequence. Cistron is a coding sequence, but the functional unit of the DNA molecule that codes for the particular gene product. Okay, so that codes for the particular gene. The correct option is option B. Next question. Read the statements given below and identify the incorrect statement. The human genome contains 3164.7 million nucleotides. So this question is related to the Human Genome Project. Okay. The average gene consists of 30,000 base pairs. The total number of genes estimated at 30,000. Chromosome Y has 231 genes. Less than 2% genome codes for the proteins. Yes, which one is the incorrect statement? You have to find out. The human genome contains 3164.7 million nucleotides. Yes, that is correct. The average gene consists of 3000 base pairs. Sorry, 30,000 base pairs. So this one is wrong. So the average gene consists of only 3000 base pairs. It contains only 3000 base pairs. So option B is the should be corrected as like this. The total number of genes is estimated at 30,000. Chromosome Y has the 231 genes. That is also correct. Less than 2% of the genome codes for proteins. That is also a correct sentence. Option B is the wrong answer. Next question. The coding sequence found in a split genes are operons, introns, exons, cistrons. So the coding sequence, they are called as exons. So these exons, they are the coding sequence present on the split genes. These introns, so they are present on the HN-mRNA. Here, so these introns, they are the non-coding sequences. So these introns are removed during the splicing process. After the transcription, splicing process will be done. So in this splicing process, introns are removed. And cistrons, you know that, so they are the coding sequences, they code for the particular gene. And operons are the, so that is the not a split genes. Next question. The removal of the enzyme affects the synthesis of the hnRNA in eukaryotes. So, which affect the formation of the hnRNA, that is the precursor mRNA, alva. So, here option A, RNA polymerase 2, RNA primase, RNA polymerase 3, RNA polymerase 1. So, which one is the correct answer here? The RNA polymerase 1 transcribe the rRNA, whereas RNA polymerase 3 is responsible for the transcription of the tRNA, 5 srRNA and small nuclear RNAs. So option RNA polymerase 1 is not correct answer, or RNA polymerase 3 is also not correct answer because they transcribes so these RNAs. But here, what is the question? Which affect the synthesis of hnRNA, that is RNA polymerase 2. Okay, the correct answer is RNA polymerase 2. Next question. Sickle cell anemia is caused when a valine is replaced by glutamic acid in the beta polypeptide chain. When glutamic acid is replaced by valine in beta polypeptide chain. When glutamic acid is replaced by valine in a alpha polypeptide chain. Then when valine is replaced by glutamic acid in alpha polypeptide chain, option D, that is. So here you know that the sickle cell anemia in the principles of inheritance and variation, you know about the how it is caused, everything. Here the it is caused by glutamic acid which is replaced by the valine. Glutamic acid is replaced by the valine, but in alpha polypeptide chain or beta polypeptide chain, it is at the alpha polypeptide chain. The glutamic acid is, beta polypeptide chain. Sorry, not alpha polypeptide chain, beta polypeptide. Here, glutamic acid is replaced by valine in beta polypeptide chain. Next question. Which mRNA will be translated to polypeptide chain containing 8 amino acids? So, which of the following contain the 8 amino acids? You know that. So, these codons, they three triplet codons, they codes for one amino acid. Okay. So, let us check this. Here in the option A, AUG. So, this is one amino acid. UUA, 2 and 3, 4, 5. UAG is the stop codon. 6, 7, 8. Okay. So here total 1, 2, 3, 4, 5, 6, 7, 8 amino acids are there. So check this one. 1. But here in this chain there is a stop codon UAG. After that there is no synthesis of the chain, polypeptide chain. So only 1, 2, 3, 4, 5 are there. 5 amino acids are there. Okay. So option A is the wrong answer. Check the second one. 1, 2, 3, 4, 5, 6, 7. So this is 8. Total 8 is there. So here is the stop codon. Next, check it once. 1. So this is 2, 3, 4, 5, 6, only 7. So this UH is the stop codon which do not codes for any amino acid. So check the last one, then it is also a wrong answer. AUG 1, 2, 3, 4, UAA. So it is the stop codon. So it does not translate after this. So stop codon is there. So only 1, 2, 3, 4 amino acids are there. So then which one is the correct answer? So this one is wrong. 1, 2, 3, 4, 5, 6. Only 7 amino acids are there. Here also not 8 amino acids. So here also not 8 amino acids because stop codon is present here. So in the option B, we can see that 8 amino acids. 1, 2, 3, 4, 5, 6, 7, 8. And at last stop codon. ಗೊತ್ತಾಯ್ತಾ? ಸೊ ಯಾಕೆ ಉಳಿದಿರೋ ಆಪ್ಷನ್ ಇಲ್ಲ ಅಂದರೆ ದಿಸ್ ಯು ಎ ಜಿ ಯು ಜಿ ಎ ಆ್ಯಂಡ್ ಯು ಎ, ಸೊ ದೀಸ್ ಆಲ್ ಆರ್ these are the stop codons. Next question. Arrange the following events in the order of synthesis of protein or in a translation process. Okay, how the protein is synthesized in a translation process. Yes, a peptide bond forms. A tRNA matches its anticodon to the codon in a site. The movement of second tRNA complex from A site to P site. The large subunit attaches to the small subunit and initiator tRNA fits in the P site. A small subunit binds to the mRNA. The activated amino acid tRNA complex attaches the initiation codon on the mRNA. Yes, which one is the first step in these all? So first step will be a small subunit. You know the translation process. I hope translation process ಅಲ್ಲಿ ಯಾವ ತರ step by step ಆಗತ್ತೆ ಅನ್ನೋದನ್ನ ನೀವು ಇಲ್ಲಿ ಬರಿಬೇಕು. A small subunit binds to the mRNA. This is the first step. Then the activated amino acid tRNA complex attaches to the initiation codon and on the mRNA. The activated amino acid, so that is formed by the tRNA. Alba. So a tRNA activated amino acids ಏನಾಗುತ್ತೆ that binds the initiation codon that attaches to the initiator codon of the mRNA. mRNA ಗೆ bind ಆಗುತ್ತೆ ಅಲ್ವಾ? Then what happens? The large subunit attaches to the small subunit and initiator tRNA fits in the P site. So that is the next step. Then a tRNA matches its anticodon to the codon in a A site. So the tRNA that is matches its anticodon to the codon in the A site. Then a peptide bond forms between them. Last step is this one 3. The movement of second tRNA complex from A site to the P site. So next they will move to the A site to the P site. This is the translation process stepwise. So first step is fifth one. So fifth one is observed in the C and D. See the next step. Next is the fourth. Alva fourth step nodi. Alla sixth. The activated amino acid tRNA complexes attaches to the initiation codon. So which one is the correct answer? I think option D is the correct answer. Yes, option D is the correct answer. That is fifth one, sixth one, fourth one, second one, first one, and the third one answer. Okay. Next question. Select the incorrect statement out of the 5 given below about lac operon when the lactose is present in the medium. When the lactose is present in the medium, gene A, that is lac A, gets transcribed into mRNA, which produces the beta galactoside and permease. Then inducer repressor complex is formed. Lactose inactivates the repressor protein. RNA polymerase transcribe Z gene, Y gene, and A gene. Allolactose is the inducer of the lac operon, which is related to this question is related to the lac operon. Lac operon ಅನ್ನ ಕರೆಕ್ಟ್ ಆಗಿ ನೀವು ಅರ್ಥ ಮಾಡಿಟ್ಕೊಂಡಿರ್ಬೇಕು. When the lactose is present in the medium, when the lactose is absent in the medium. So both in the both condition ಏನಾಗತ್ತೆ ಅನ್ನೋದನ್ನ ನೀವು ತಿಳ್ಕೊಂಡಿರ್ಬೇಕು. Here incorrect statement ಯಾವುದು? So the lac A gets transcribed into mRNA, which produces the beta galactosidase and the permease. So that is the wrong answer. Lac A that produces transcribed into mRNA, which produces only beta galactosidase, not produce the permease. So that is the correct answer. Next question. The percentage of human genome which encodes the protein is approximately less than 2%, B 5%, C 25%, D 99%. The percentage of human genome which encodes the proteins approximately, that is less than 2%. So that is in the Human Genome Project. Okay. The correct answer is option A. The enzyme which can break down the seal and seal the DNA strand. Here, which break and seal the DNA strand, which is the correct answer for this question. The DNA strand is, so that is broken by that is separated by the enzyme helicase. Okay, the helicase enzyme that separate the DNA strands like this. So that is able to form the separate strand of the DNA strand, single strand DNA, and seal the DNA strand, which can uh, break and seal the DNA strand. That is restriction endonuclease enzyme. So which break the DNA, and again it is sealed. Okay. Next question. Which of the statements given below is correct with respect to the frame shift mutation? A single nucleotide base change, insertion or deletion of the genetic material. Glutamine is replaced by valine. This is not a correct answer. Glutamic acid is replaced in a valine. So that is in case of the sickle cell anemia. Sickle cell anemia is example. That is not a correct answer for the frame shift mutation. Sickle cell anemia is the example for the point mutation. Alva, point mutation is the example. Insertion or deletion of a number of nucleotides in a DNA sequence that is not divisible by 3. Insertion or deletions of a number of nucleotides in a DNA sequence that is not divisible by 3. That is the option D is the correct answer. Next question. Transcription initiation factor associated with the RNA polymerase holoenzyme in prokaryotes. So in prokaryote, so that is associated with the beta factor, omega, sigma or alpha. What is the correct answer? So which factor is associated with that? So that is also called as the initiation factor. Here, which is called as a initiation factor. So option C, that is sigma. So this sigma is called as a uh, the initiation factor. The stretch of codons between AUG and stop codon is called the AUG and the stop codon. What they are called? Open reading frame, data box, collinearity and degenerate. What they are called? They are called as open reading frame. These codons are called as open reading frame. Next question. The structural genes of lac operon transcribe the mRNA which is polycystonic, replicative, monokaryotic and monocystonic. Here option A is the correct answer. The structural genes of lac operon transcribe the mRNA which is polycystonic. Option A is the correct answer. Next question. If the sequence of bases in DNA, that is TAA, CCGA, CCA, then the sequence of codons on the transcript will be. So codons on the transcript means in the mRNA ಅಲ್ವಾ. So mRNA ಮೇಲೆ ಏನಿರುತ್ತೆ, so that mRNA, just for example, this is 3 prime to the 5 prime. The mRNA will be 5 prime to the 3 prime direction, and thymine, so that is adenine, here guanine, just sorry, uracil. Thymine is replaced by uracil, so AT is not correct answer alva. Then guanine, AUG, then CG alva. Then GA is there, so this is the wrong answer, option C. Illodi, the just find out the correct sequence. So this is the DNA strand. This is the DNA strand. Then the sequence of codons on the transcript will be transcription. So a transcript means on the mRNA. Transcript, that is a mRNA ಅಲ್ವಾ. mRNA ಮೇಲ್ಗಡೆ ಯಾವ ತರ ಕೋಡಾನ್ಸ್ ಇರುತ್ತೆ ಅಂತ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ. ಸೊ ದ DNA strand, so that act as a 3 prime to the 5 prime, that act as a template strand to synthesis of the mRNA, and mRNA always will be in a 5 prime to the 3 prime condition. So here this thymine, that is replaced by the adenine, ATCG. Always adenine binds with the thymine, cytosine CG. So adenine, thymine, but in case of the RNA, thymine is replaced by the uracil ಅಲ್ವಾ. uracil ಜೊತೆ replace ಆಗಿದೆ. ಇಲ್ಲಿ ನೋಡಿ thymine DNA strand ಅಲ್ಲಿ ಇದೆ, so adenine, that is correct answer. Adenine, thymine, T is replaced by U ಅಲ್ವಾ, uracil, so I will write U. So option B ಮತ್ತೆ option A ಅನ್ನ cancel ಮಾಡ್ತಾ ಇದ್ದೇನೆ. Then C, so that is G. Again C is there, G. Again G, so that is C. ಅಲ್ವಾ ನೋಡಿ, AUG, G, AUGG, CU, AUGG ಮತ್ತೆ A ಬಂದಿದೆ, so this is not a correct answer. Option C is the correct answer. I hope this question is clear. Next question. Next, find the match the following in the given column 1 with the column 2. Okay. So here in the column 1, enzymes are given. In the column 2, so their functions are given. Okay. So what you have to do, you have to match the column 1 with the column 2. ನೋಡಿ, helicase enzyme. What is the function of the helicase enzyme? That unwinds that break down the hydrogen bond. ಇಲ್ಲಿ ನೋಡಿ, so this is the, just imagine this is the double strand DNA which break down the hydrogen bonds between the two strands and unwinds the DNA strand ಅನ್ನ separate ಮಾಡುತ್ತೆ ನೋಡಿ. Activation of amino acids, joins the DNA fragments, unwinds the DNA helix. So option A, O, that is the correct answer. Then which one you know? DNA ligase that act as a molecular glue ಅಲ್ವಾ. ಅದೇನ್ ಮಾಡುತ್ತೆ, bind ಮಾಡುತ್ತೆ, joins the DNA fragments, that is DN. So let us check with the option. Option A, O ಅಲ್ವಾ. Option A, O ಇದೆಯಾ ನೋಡಿ. Option A, O ಇದೆ, but A R ಇದೆ, wrong one. A M ಇದೆ, wrong one. A R ಇದೆ, so wrong one. So we can easily find out the answer A O. And check with the DNA ligase, joins the DNA fragments, DN. Yes, DN is also correct. Then DNA polymerase, So what is the function of the DNA polymerase? That is DNA synthesis, which add the nucleotides and synthesis of the DNA. That is also correct answer. Then B, peptidyl transferase. So that is forming the P, uh, peptide bonds between the amino acids, which forms the peptide bond between the amino acids. Yes, that is the correct answer. Then amino acyl synthetase enzyme. Amino acyl synthetase enzyme, that is activation of the amino acid. So here option A is the correct answer. Then RNA primase, synthesis of the RNA primer. So that is the option A. Option A ಏನಾಗಿದೆ. So that is the correct answer. Next question. Which of the following triplet codon do not code for any amino acids? Which among them? So AUG is the, so that is the initiator codon, UAA, CCC, GAU. So here UAA, which do not code for any amino acid because it is called as a stop codon. Stop codon anta karitare adrinda, so that is that do not code for any amino acids. Next question. DNA dependent DNA polymerase catalyze the polymerization in the direction of, Nodi, there are two DNA molecule, DNA strands. One strand that is having the 3' to 5' direction, and another strand that is having the 5' to 3' direction. So there are two strands, but here always the synthesis of the new strand that begins with the, always with the 5' direction. The new strand that synthesizes in the direction of 5' to 3' means in this direction. So e directionally synthesis ಆಗತ್ತೆ. So that is the 5' to 3' direction. Next question. Polymerization by DNA polymerase is in. So that is also same question. Polymerization of DNA polymerase. DNA polymerase ಏನ್ ಮಾಡುತ್ತೆ? That adds the nucleotides during the synthesis of the new strand. ಅಲ್ವಾ? ಇಲ್ಲಿ ನೋಡಿ, I will take, this is the DNA replication fork. Just a minute. So DNA, in our DNA replication process, so this is 3' to the, sorry, 5' to 3' direction, and this is 5' to 3' direction. The new strand synthesis, that is always, so that is having the 5' to 3' direction. So it start with here 5' to the 3' direction, because so both are they should be in a opposite polarity alva. Polarity ಯಾವ ತರ ಇರಬೇಕು ಅಂತ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ. So that is the correct answer is option B, that is 5' to the 3' direction only. ಯಾವಾಗಲೂ ಏನಾಗುತ್ತೆ? 5' to 3' direction ನಲ್ಲಿ synthesis ಆಗುತ್ತೆ. Next question. The introns are removed and exons are joined. Define. So just a minute. The introns are removed and exons are joined in a defined order is capping, splicing, tailing, replication, fork. Here, capping, splicing is the correct answer here. So hnRNA, that is the precursor of mRNA. So just imagine this is the mRNA. So here, so these blue color segments, they are the introns, means they do not code for any amino acid. So here they do not code, so they are not functional. So these are the exons. So these introns are removed. So these introns are removed and only exons are joined. So these exons are joined. So that is called as a splicing process. So after that, capping and tailing. So adding the some other things to the that exon. So that is forming the mRNA. So the correct answer is splicing. What is capping? What is tailing? So that I will explain in the next 2 mark question. Okay. Next question. Triplet codon refers to a sequence of 3 bases on mRNA, tRNA, rRNA, none of this. The triplet codon, that is AUG and GUA. So like this UAA. So these are the codons. So these triplet codons, where they are present? They are present on the mRNA. They are present on the mRNA. Option A is the correct answer. Next question. The number of nucleotide pairs present in the DNA of primary oocyte of newborn human is, that is 3.3 into 10 raised to 9, 6.6 into 10 raised to 9, 13.2 into 10 raised to 9, 3.3 into 10 raised to 7. Here, the number of nucleotide pairs present in the DNA of the primary oocyte, so of a newborn human, is 3.3 into 10 raised to 9 base nucleotide pairs. In lac operon, if mutation occurs in the Z gene, trans acetylase will not synthesized, beta galactosidase will not synthesized, permease will not synthesized, lactose digestion will be rapid. Here, so in a lac operon, ZYA, they are the structural genes. Here, Z that transcribe the, so beta galactosidase ಅಲ್ವಾ ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸೈಡೇನ ಸಿಂಥಸಿಸ್ ಮಾಡುತ್ತೆ. ವೈ ದಟ್ ಸಿಂಥಸಿಸ್ ದ ಪರ್ಮಿಯೇಸ್ ಆ್ಯಂಡ್ ಎ ದಟ್ ಸಿಂಥಸಿಸ್ ದ ಟ್ರಾನ್ಸ್ ಅಸಿಟೈಲೇಸ್ ವಿಚ್ ಸಿಂಥಸಿಸ್ ದ trans acetylase. So here trans acetylase will not be synthesized. So that is not a correct answer. Beta galactosidase will not be synthesized. Zyzine that is responsible for the synthesis of the beta galactosidase. Permease will be synthesized by the Y, and lactose digestion will be not rapid. So that is also a wrong one. The correct answer is beta galactosidase will not be synthesized. Next question. A DNA segment has total 1000 nucleotides. Out of 240 of them are adenine containing nucleotides. How many pyrimidine bases this DNA segment possess? Okay. Here, adenine, adenine, adenine binds with the thymine. Cytosine binds with the ATCG. Alva, cytosine. Here, adenine and guanine, they are purines. And cytosine and thymine, they are pyrimidines. Okay. Here, 1000 total number of nucleotides, that is 1000. Out of 240 of them are adenine. 240 is adenine. Then what is the thymine number? That is also 240. So 240, 240. So that is 480. Alva, 480. Out of 1000, out of 1000, 480. That is 0. 2, that is 520. 520 remaining. So here in the 520 divided by 2, because so cytosine and guanine, they are in the same number. So total 520 is there. So that is 260 plus 260. Then how many pyrimidine bases this DNA segment possess? So how many pyrimidines this DNA possess? So that is 500. ನೋಡಿ ಇಲ್ಲಿ ಅಡಿನೈನ್ ಮತ್ತೆ ಗ್ವಾನಿನ್ ಅನ್ನ ಪ್ಯೂರಿನ್ ಅಂತ ಕರೀತಾರೆ, ಸೈಟೋಸಿನ್ ಮತ್ತೆ ಥೈಮಿನ್ ಅನ್ನ ಪಿರಿಮಿಡಿನ್ ಅಂತ ಕರೀತಾರೆ ಅಲ್ವಾ. ಹಿಯರ್ ಅಡಿನೈನ್ ದಟ್ ಈಸ್ 240 and guanine that is 260 ಅಲ್ವಾ. ಬಟ್ ಹಿಯರ್ ATCG ಅಂತ ಇದೆ. ಅಡಿನೈನ್ ಬೈಂಡ್ಸ್ ವಿತ್ ದ ಥೈಮಿನ್, ಸೈಟೋಸಿನ್ ಬೈಂಡ್ಸ್ ವಿತ್ ದ ಗ್ವಾನಿನ್. ಇಲ್ಲಿ ಪಿರಿಮಿಡಿನ್ಸ್ ಎಷ್ಟಾಯ್ತು? ಪಿರಿಮಿಡಿನ್ಸ್ ಆಲ್ಸೋ 240 plus 260, that is total 500. So the correct answer is 500. Which of the following is a stop codon? ಇದರಲ್ಲಿ ಸ್ಟಾಪ್ ಕೋಡಾನ್ ಯಾವುದು? AUG is the initiator codon. GUG, UAG, CAU. Here the correct answer is option C, UAG. UAG is also a stop codon. UAG also a stop codon. Okay. So till now we have discussed some MCQ question. Let us start the 2-mark question discussion. Okay. So this is also one of the very important section. So where 2 marks question will be asked. Okay. So let us discuss the 2-mark question. So this question that is from the model question paper. Okay. Model question paper. Here the first question. Write a note on role of sigma and rho factor in transcription in the prokaryotes. What is the function of the rho and sigma factors? Here, the sigma factor, so that is called as, just a minute. So the sigma factor that is called as a initiator factor. So that is the initiator factor which initiate the transcription, where the rho factor that is called as a terminator factor which which ends the transcription. So this much is enough for the 2 mark. So what is the function of the sigma and what is the function of the rho factor? Mention the levels where gene expression can be regulated in eukaryotes. Here the answer is transcription level, how the gene expression can be regulated. So in the transcription level, processing level, so regulation of splicing, transport of the mRNA from the nucleus to the cytoplasm, and the translation level. So in this level, the transcription or the gene expression can be regulated in eukaryotes. Okay, one of the very important question. Next question. Which sequence of the bases transcribed from DNA are found both in hnRNA and mRNA? Here, the sequences which are transcribed from the DNA that transcribed into hnRNA. So that is the precursor of the mRNA. Then after that it will become mRNA. Alva. So here the DNA, the sequence bases transcribed from DNA which are present in both in hnRNA and mRNA, that is nothing but the exons. Exons are the sequences, so that is transcribed into mRNA and ultimately translate the proteins. So they are present both in the hnRNA and mRNA. While introns ಏನ್ ಮಾಡ್ತಾರೆ hnRNA ಏನಿರುತ್ತೆ, so that contain the non coding sequences, that is called as a introns ಅನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ ಸ್ಪ್ಲೈಸಿಂಗ್ ಅಲ್ಲಿ ರಿಮೂವ್ ಮಾಡ್ತಾರೆ. ಬಟ್ exons are the coding sequences which are present on both hnRNA and the mRNA. Next question. Draw a diagrammatic sketch of lac operon when the lactose is present in the medium. Very important question. ಈ lac operon concept ಇದೆ ಅಲ್ವಾ, so that can be asked as a 5 mark question. Also it can be asked in the 2 mark multiple choice question. Be clear with that topic. One of the very important topic here. They ask the draw the diagrammatic sketch when the lactose is present in the medium. Lactose present ಇರಬೇಕಾದ್ರೆ sketch ಮಾಡಿ ಅಂತ ಡ್ರಾ ಮಾಡಿ ಅಂತ ಅಷ್ಟೇ ಕೇಳಿದರೆ. Sometimes they will ask the explanation also. So this is the diagram when lactose is present in the medium. Okay, so here when the lactose is present, the repressor gene that produces the repressor mRNA, and it produces the protein, and repressor protein produces the that bind with the inducer. Inducer is nothing but the lactose here. Then this repressor protein that is inactivated, that is unable to bind the operator alva, operator anna bind madodilla. Then so this structural genes they are able to transcribe, and they transcribe the mRNA, and they transcribe the beta galactosidase, permease and transacetylase alva. So this is the process when in the presence of the inducer, that is lactose. Why capping and tailing is required from the for the hnRNA? Here the capping is the unusual nucleotide where methyl guanosine triphosphate is added at the 5 prime end of the hnRNA. So I told you that I will explain what is capping and what is tailing. Here what is capping? Methyl guanosine triphosphate that is added at the 5 prime end of the hnRNA. And what is tailing? Tailing is the adenylate residues, that is 200 to 300. So that are added at the 3 prime end of the template hnRNA, that is called as a capping and tailing. Why it is required? It is a process. It is the fully processed hnRNA. Just a minute. So here it is the fully processed hnRNA, now called as a mRNA. So that is transported out of the nucleus for the translation process. The fully processed mRNA is formed by the capping and tailing process. Okay. So these are the some important questions. So we have discussed in this session. So in the next session, we are discussing some more important 2 mark question as well as 5 mark question. Okay. Thank you. We'll continue in the next session.